Here is Ma Xianyun, and I will talk about lab on a CD. Off-center microfluidic biochip or off-center microfluidic biodisc is a lab-on-a-chip technology, also known as a lab disk, that can be used to integrate separation, mixing, reaction, and other processes and detect molecules in a single platform, including optical disc or DVD. It is also known as laboratory disk, LOD, systems and includes subcategories of laboratory on-chip, LOC, devices. Advantages of the LOC platform include reduced cost, use of fewer materials and reagents, faster reaction times due to small liquid volume and diffusion distance, portability and programmability. Recent developments have made CD microfluidic technology increasingly portable, low cost, and easy to use, making it an ideal system for point-of-care applications. The lab-on-a-chip advantages are reduced material cost, reduced reagent volume and cost, faster mixing, portable and programmable, time-saving and labor-saving. The disadvantages are tubing is required for fluid pumping, difficulty handling larger volumes, bubble formation, difficulty of multiplexing. The lab on a disk advantages. The lab on a disk advantages are uses only a motor to pump fluids, allows for a closed system, pumps all fluids regardless of properties, able to work with a wide range of volumes, multiplexing of several assays on one disk, easy to mix and meter fluids, density-based separation, eliminates trapped bubbles. The disadvantages are unidirectional nature of liquid flow and difficulty working with very small volumes. The main driving force of the fluid in a disk is centrifugal force, which pushes the liquid radially outward from the center of the disk, forcing it through channels and chambers. The centrifugal force acting on a particle, or liquid per unit volume, is shown as follows. Where ρ is the density of the liquid, r is the average distance from the liquid to the center of the disk, and omega is the angular velocity in rad/s. The successful adoption and use of LOD systems depends on inexpensive and reliable manufacturing solutions for disposable. Common materials used in microfluidics disks include, but are not limited to, polycarbonate, poly-methylmethacrylate, PMMA, cyclic olefin polymer, COP, polydimethylsiloxane, PDMS, and polyurethane. Microfluidic disks are typically made of adhesive layers and hard plastic layers for rapid prototyping purposes. Effective valve technology is at the heart of sample-to-answer analysis, maintaining liquid volume isolation from the rest of the system during operations such as sample cracking and mixing, allowing for accurate equalization, and preventing liquid leakage during temperature changes. Valve technology on off-center microfluidic platforms can be divided into three different categories. Active valves require external actuation and are therefore more reliable and robust than passive valves. Passive valve actuation depends on the interaction between surface tension and centrifugal force acting on the liquid in the plate. Semi-active valves are angular velocity-related valves that provide a higher level of valves, but they are simple to manufacture and do not involve surface treatment. In biological and chemical analyses, the original sample must undergo a series of preparatory operations, which may include cell sorting or sample concentration, lysis of cells for analysis requiring genomic material, sedimentation to separate any sediment from the separation. Here is typical molecular diagnostic assay. Particle sorting is required when the number of target cell types in the cell population is small. Sample purification and concentration are used to detect low concentrations of components. Sedimentation filtration is the determination of fluid by removing solid components. Cell lysis is the extraction of genomic or proteomic material from bare cells. Nucleic acid, Na, amplification is an integral part of genetic analysis and can detect copies of several biomarkers. The LOD system uses two analytical methods, optical and electrochemical. Optical detection has fluorescence detection and colorimetric detection. Electrochemical detective alternative to optical detection because of its low cost, small footprint of equipment, high sensitivity, specificity, and portability. There are ampere detection and onboard flow monitoring. This article is a summary of LOD molecular diagnostic systems. The technology is complex, but more and more people are trying to overcome it. With the technology, it is possible to effectively store samples and analyze samples in a variety of environments.